My name's Adrian Randolph, Dean of Weinberg College of Arts and Sciences, and I'm delighted to welcome you here this evening. I'm going to say a few introductory remarks, and then my colleague Ravi Allada is going to come up and introduce our speaker more formally, and then he will come to the podium. So I'll start with a quotation. Time, likewise, does not exist per se, but the census arises from things themselves, namely from what has been in the past, then what thing is present, further what follows next. Nor must it be confessed that anyone perceives time per se, separated from the motion and calm rest of things. I'm an art historian, and I work on the Italian Renaissance, and I started out as a classicist, so I occasionally draw on my pool of knowledge when I'm trying to deal with things that are far beyond my abilities, and that certainly counts tonight. That was an excerpt from the first century writings of Lucretius, whose De Rerum Natura forms the basis of much of the assumptions we make in modern science. Certainly, I would imagine our speaker tonight would phrase things rather differently, but I think Lucretius pointed out a path which we find ourselves on today, and not least our esteemed speaker finds himself on today, by looking to time not only as a record of things that are fixed in some planetary gyrations, but rather a census, that sense of being in the world, and in his work, lodged in the molecular, lodged in the DNA, lodged in the things, as I said before, I can't possibly understand. To those of us who've lived through the revolution set in motion by those names Watson, Crick, and Franklin, and the discoveries that occurred in the vortical wake of the discovery of the double helix, we've witnessed the fetishization of the mysteries of DNA. Not only do many of us remember high school classes trying to line up those dreaded sort of alphabetical lines between things that I fully didn't understand, but it's become part of the koine, part of the way we speak. We always talk about things being in our DNA. Indeed, even at this institution, occasionally we trot out the idea that "and" is in our DNA, which points not only to something perhaps germane about Northwestern, but more relevantly, I think, to the way in which genetics have become part of our everyday way of thinking about ourselves. Now, there is, I understand it, a subculture in genetics, the RNA-ophiles, who follow not the reproductions of and the copies of material per se, but rather its conveyance around that sort of miasmic liquid, which I assume inhabits all our bodies. And at the heart of this conveyance, the scriptorium of the human and the animal, I suppose, and the biological in all forms, is messenger RNA. And again, I have no idea what that is, but I take it that it is germane to Michael Rosbash's work. And I hope I'm pronouncing your name right. I didn't ask you that before. If Michael Rosbash is appropriate, good. So Michael, I'm sure, has had occasion to muse at one point or another on the passage of time. He works with generations of fruit flies, hundreds, millions of generations, I assume, by now. And not to mention the rhythms of time in these animals, these dew-loving little flies that are the basis of so many scientific discoveries. I think we should pay homage to this tiny creature, which I take it is leading the way again in scientific discovery. So I won't say much about Michael because Ravi's going to introduce him, but I want to say thank you very much for coming to Northwestern on behalf of the institution, the president, the provost, board of trustees, any other dignitaries we care to mention. Thank you for joining us, and congratulations on your most recent, perhaps most recent award, I don't know, certainly the one that distinguishes you much in the world's eyes. And now, before asking Ravi and then Michael to come up, I just want to say a few words about Nina Schwartz. And I realize this is sort of an interruption to the normal sort of state of affairs. Some of you will know that our esteemed colleague Nina Schwartz, William Deering Professor Emerita of Biological Sciences and in the Department of Neurobiology and Physiology, passed away recently. And I simply want to say how important her path-breaking work and life was to the life of Northwestern and so many people who work in the life sciences. And I didn't want this occasion to go by without reminding this generation, especially of the work of this esteemed woman who really fought both in the lab in wonderful ways, but also in the, let's say, the institutions of science. And I just wanted to pay homage to her life and work before we move on to the celebratory events this evening. So thank you for joining us, Michael. Congratulations on your award, which I'll let other people speak about. And Ravi Allada, Chair of the Department of Neurobiology, will now come to the podium. Thank you.
All right. Thank you, Adrian, for that fantastic introduction, really. It is my great honor to introduce our speaker today, Michael Rosbash from Brandeis University and the Howard Hughes Medical Institute. And I should say first that when I extended this invitation, this actually predates the awarding of the Nobel Prize. So as we would say in the biological clocks field, excellent timing. It was the work of Seymour Benzer and colleagues that discovered that single genes could actually play a major role in animal behavior. And he used the aforementioned fruit fly, Drosophila, as a model system. And it is really Michael's pioneering work that allowed us to go from that mutant fruit fly that Benzer and his colleagues had found to actually understanding the molecular and neural basis of behavior. And he used as a model system circadian rhythms, or the daily rhythms of our own sleep-wake cycles. Now, as one of the early pioneers in the field of molecular biology, Michael and his colleagues were the first to clone a so-called behavior gene, the period gene, or PER for short. And it was the discovery of PER, the cloning of PER, that allowed the finding that PER was a core component of the gears of a circadian clock, and that these gears consisted of a transcriptional feedback loop, one in which the PER gene is transcribed into the aforementioned messenger RNA, and then feeds back at the protein level to control the synthesis of its own messenger RNA or its own transcription. Now, since the discovery of these transcriptional feedback loops now over 30 years ago, this idea has really stood the test of time. It's extending to independently evolved clocks in plants and fungi. Its core components are conserved even with our own human circadian clocks. It's present in virtually all of our organs and tissues, and it regulates at least half of our genomes. So it's really not an understatement to say that as a result of this work, and I paraphrase the evolutionary biologist Dobzhansky, that nothing in biology makes sense except in light of time. So Michael has made many more contributions. I could go on and on. He's made some very important discoveries related to the neural basis of that circadian sleep-wake cycle in the fly. But I think I'll try to keep it short to give Michael some time to give his talk. Now, his discoveries have been recognized by too many awards and honors really to list, but just to name not so few. Election to the National Academy of Sciences, the Gruber Prize, the Canada Gairdner International Award, the Massry Prize, the Wiley Prize, the Shaw Prize, and this last fall, the Nobel Prize in Physiology or Medicine. So please join me in welcoming Michael Rosbash.
Thank you very much for the invitation. It's a pleasure to be here. As Ravi said, this was extended before October 2nd, and I'm loath to use that event as an excuse to not appear somewhere, and especially with a former trainee and now a close colleague and even friend. Ravi, it becomes impossible. Your remarks, the dean's remarks, reminded me of my co-winner and longtime friend and colleague Jeff Hall at Brandeis, who would often say, after remarks such as yours, he would turn to me and say, "I didn't understand a single word he said." And that's really not true. I understood about half of what you said, and I was grasping when we met. And for 10 seconds, I was grasping at a connection, as you saw, with the Italian Renaissance. And I will say that Stephen Greenblatt is my high school classmate. So we went to high school together. Lucretius is a sort of interface there. Philistine though I am. So here we are. So let me begin, and a quarter of you know all this intimately well, but 3/4 not so well. And so let me just say that circadian rhythms arose in response to this inexorable turning of the Earth, a light-dark cycle that appears every day, changes in illumination and temperature. And this environmental cue preceded the current constitution of the atmosphere, nutrition in any form similar to the way it is today for any animal whatsoever. And so this really is a cue which has been there for so long that it turns out to be really good for organisms to adapt to. And as a consequence, these rhythms in one form or another, a daily timer, are nearly ubiquitous. And the illustration on the bottom going from your left to right is cyanobacteria who are carrying a reporter gene which is high, gives off light at one time of day, and then low illumination at another time of day. In the middle is a very famous rhythm, ostensibly the first one to be recognized and experimentally addressed, namely leaf movements of plants which extend their leaves at times when photosynthesis should be maximal, and then retract the leaves to prevent water loss and respiration and so forth and so on at night. And on your right are two examples of slices from the suprachiasmatic nucleus, which carry a reporter gene, again, a bit like the cyanobacteria on the left, which are illuminating at one time of day, a little bit of illumination other time of day, so this inexorable beating of the clock. And so the properties that we're interested in, if you will, or the properties which define the clock and also the purpose of the clock are that these rhythms are self-sustained. That is, that they continue in constant darkness or under constant conditions, but they entrain to these light or temperature cycles. So in fact, for us humans, we have a endogenous clock of about 24 hours and 15 minutes. Some early birds a little bit shorter, some night owls a little bit longer, but by and large, 24 hours and 15 minutes. And the reason that tomorrow is like today and the day after is like the day before is because the light-dark cycle entrains and then shifts the clock every day. So we have a not very accurate timer. It's not perfectly 24 hours, and then every day, dawn, if you will, resets the clock. And the purpose of this machine is to anticipate what's going to happen. The early bird gets the worm, and the early worm avoids being eaten by the bird. And secondly, somewhat less certain from an experimental point of view, that probably the clock is governing internal events, keeping different reactions separated temporally, so that reactions, for example, that like oxygen are separated from reactions that don't like oxygen when oxygen is high or low in the bloodstream, et cetera. So I'm going to say very little about this prize other than one or two little snippets. A week after this event was announced, I went to dinner with friends who had a long-standing, like this visit to Northwestern, a long-standing dinner obligation with close friends of ours, and this was the dessert they produced, and this fantastic bakery made this design there. And this is quoting me at my press conference at Brandeis a few hours after the announcement. It's a great day for the fruit fly, and of course, Jeff Hall, as I said, my colleague, and Mike Young at Rockefeller, the co-winners. We're really quite honored by this, flattered, surprised, all of the above. And to put this in perspective, for those of you who don't have an appreciation for this humble organism we work on, this is the 5th Nobel Prize for the fruit fly. And this began with T.H. Morgan, the genetic pioneer, in 1933. He was not only the first, let's say, fruit fly winner, but he was the first American to win the Nobel Prize. A fact I learned at the Swedish embassy in November when they had an event for us. I had no clue that he was the first American. And then Hermann Muller, Nusslein Volhart, and Eric Wieschaus, who are still around, and active Drosophila scientists, along with Ed Lewis, who passed away. And finally, I think it was 2011, if I'm not mistaken, Jules Hoffman, who shared the Nobel Prize on innate immunity, and we are the fifth. And I should say one of the perks, one of the several perks, along with throwing out the first pitch at Fenway Park 3 days ago, that this prize brought me was a date on Wait, Don't Tell Me. And Paula Poundstone, who was really smart, when I said on the radio show, I said, this is the fifth Nobel Prize for fruit fly, she said to me, those poor animals, 5 of those heavy metals around their neck, how do they possibly manage? Anyway, so there we are. So fruit flies and us, right? This is a quick and incomplete summary of the hormones and organs that are conserved between humans and flies. And you all see, or you should see, things down there as biologists that you would be interested in. And the organ systems are sort of cartooned on the 2 sides of the slide. I'm not going to belabor that, of course. Human disease genes, the genes that I was about to say we all know and love, genes that are incredibly important for disease and also for basic research that have given us insight into all kinds of pathways. 80% of these genes are conserved between humans and fruit flies and can be, if you'll forgive me, fruitfully studied in Drosophila. And just to list a small subset of the diseases that are under study, some of which here at Northwestern, cancer, neurodegenerative diseases, Huntington's, Alzheimer's, drug addiction. You feed flies ethanol, they really behave like alcoholics. Obesity, diabetes, and so the list goes on and on and on. And so we're talking about an organism that not only is important for research, but as I was discussing with Tiffany today, she has fruit fly envy for the cost of that animal compared to mice. Much cheaper, short generation time, et cetera. So that's my introduction, if you will, and I want to tell you now the sort of a bit about this circadian story, and Ravi gave it to you in words, but I will start again from the beginning and try and fill this out for you. Why did we win this thing, right? And it does begin, as Ravi said, with Konopka and Benzer. Seymour is on your right, and Ron Konopka, not as the student he was when all this began in the late 60s, but more recently is seated next to Seymour. And this illustrates the phenotypes that they identified in these 3 mutants of the period gene. And for those of you who know a drop of genetics, these were 3 alleles of a single gene. And the remarkable thing was that one of them sped up the clock and one of them slowed it down. And then one of them eliminated the clock altogether. All single point mutations, single nucleotide changes, single amino acid changes, or a stop codon in the case of the null mutant, all in one gene. All that was learned many years later, but the foundation that they laid was absolutely remarkable. And this was published in PNAS. This was a top journal at the time. And for those of you who chase high-profile papers, and we're all victims of this system we live in, and so all of us have to do this to some extent, I should just point out that this paper had a total of 10 citations in the first decade after it was published. So in other words, some things, cream rises to the top. That's thermodynamics, but the kinetics is uncertain. And so it's a bit like death, right? So that's the world we live in. So those people here who were associated with the NSF Center for Biological Timing at the beginning will recognize this slide. That's where it comes from. This is a picture of a rodent who is running in a wheel. And the slide is so old, you can see that it predates modern computer technology. The recording is being done on a chart recorder. But the principle is identical, and that is that the animal loves to run on that running wheel and will do so for many hours at a time. And in constant darkness, you see the pattern of activity, which are the ticks, and the sleep time, if you will, or rest time is indicated by the long period with no ticks. And that pattern drifts to your left because the animal has a short period, an endogenous short period. So in constant darkness, the phenomenon every day is a little earlier than the day before. But the animal can entrain to a light-dark cycle. So the pattern below, the lower pattern, which says entrained, is exactly bang on the 24-hour light-dark cycle because every day the clock is shifted a bit. And you'll also notice this animal is nocturnal. That is that the activity is coincident with the nighttime, and they sleep in the daytime. So that's rodents. How do we do this? This is an assay. This is a variation on the theme that the Benzer lab, Konopka Benzer, introduced. We have single flies in these tubes, and there's food at the bottom and a stopper at the top that gives you a flavor for the size of the tube. And then those tubes are snapped into a little device. This is one of our original ones. In fact, it's the one we used at the beginning of our work at Brandeis that we gave to the Nobel Museum. One of the many things they said is you have to come with a gift. And so my wife said, why don't you give him one of your early monitors? And that woman has no end of good ideas. And so this is the tube, which is snapped into a little holder. And on either side of the middle of the tube is an infrared light source and a photoreceptor on the other side. And so this is a beam break device. Scientists forever have watched animals run around by breaking a beam. And so the flies are running up and down, and they break the infrared light, and they can't see in the infrared. So for all intents and purposes, they're in constant darkness, but we can record the events, and that's sort of how this works. And I'll show you a slight variant on this theme in a few minutes. So as Ravi said, we at Brandeis, Jeff Hall's lab and mine, cloned this gene and sequenced it in the early 80s. The work was done independently and in parallel by Mike Young's lab at Rockefeller. And we thought we'd sequence this gene and we'd know what it did, and that would be the end of the story. And of course, it was the early days of DNA sequencing. There were perhaps 50 genes in the database at the time. And they were all enzymes that people knew things about too. So it didn't match. It had no relatives in the database. And so we were stuck for about another 5 years of trying to figure out what might this protein do. And finally, thanks to Paul Harden in my lab, he discovered that the messenger RNA levels that emanated the molecules that Adrian, you spoke about, RNA. No, he's awake. He was looking at the slide. That's not fair. Those RNA molecules underwent circadian oscillations in time. And the period or phase of those oscillations was affected by the point mutants in the protein in exactly the same way the behavior was affected. And we therefore proposed that there was a feedback loop. The protein inhibited its own production, its own transcription, and that lay at the heart of circadian timing and was upstream of the behavioral phenomena which occurred in parallel. And there's a more elaborate story which I'm going to not belabor in showing over a series of 3 or 4 years and a half a dozen papers or so that general idea was correct, and the epistasis, or the order of events, from molecular to behavior was correct. And Paul really pioneered these experiments from many points of view. And he gave me the original, as a present, when he came up for the party we had at Brandeis, he gave me the original gel. And that's the original. This is before PCR, for those of you who do this kind of thing. And so this was done by RNase protection. And that's the oscillation of the transcripts which come from the transgene with 1 hour time points. You can see the transcript wax and wane there on the gel. And so we had this model. We felt we were modestly confident in it. I should say modestly, because nothing is 100% true in our business. And if you're both a good scientist and a drop neurotic, and maybe those are not separable, you always wake up at 3 in the morning, or I should say often, and wonder, how could you have screwed up? And the answer is many different ways. And so the story proceeded, and we really wanted to identify X. What's the transcription factor that's driving the production of its negative regulator, period? And this is where Ravi entered the story. He entered a couple of years before the publication. And we undertook a genetic screen to look for other mutants that might fill out the story. And we did this by looking not for period-altering mutants, by looking for through arrhythmic mutant strains. And of course, you can be arrhythmic for a gazillion reasons. That is, the downstream machinery can be messed up, motor problem. It's not very informative. But we did a subscreen looking for non-cycling and low levels of the direct products of that putative transcription factor X, so that period, and in fact its partner I haven't mentioned yet, timeless, would have very, very low levels. And we came up with the 2 key proteins, clock and cycle, as partners, which turned out to be X. And I should say that a year before, Martha Vititurna, where are you, Martha? There you are. Martha Vititurna and Fred and Joe here at Northwestern did a genetic screen in mice and identified the mouse clock gene. And this is itself a remarkable story, because the 25th mouse, if I've got it right, turned out to be the mouse with the key phenotype. So in a way, it's a common. Konopka also had an extraordinary amount of fortune in how early in the process the mutants appeared. And this not only saves you a lot of work in having to go through thousands, but it boosts your morale. We might actually be doing something that makes sense here. And the only reason why I didn't put this first, usually I present this before mentioning a lot of work from our lab, was that the initial cloning of this didn't actually show that this protein produced the negative regulators. That was done at just exactly the same time as Ravi's work at Brandeis did. So in fact, from a feedback loop perspective, the work was done at just about the same time. And so that led to this conserved feedback loop, the fact that clock and cycle, as they're called in flies, and their orthologs, clock and BMAL, in mammals, drive the transcription of these negative regulators, one of which is the same and one of which is different in the 2 systems. And orthologs is a word which means that these are not only the best matches in the 2 systems, mammals, humans/mice on the one hand, and flies on the other, but they must have been derived from a common ancestor. So in other words, these already existed 550 million years ago in the organism which was our common ancestor. That's how similar these processes are. And the dates, so this was really solidified experimentally in 1998, and the reason I have 2018 on there, other than it's today, is that this, as Ravi mentioned, this is, I think, stood more or less the test of time and is not very altered in the 20 years that have passed. And this description, this scientific sketch, if you like, is ignoring a whole bunch of other things that I'm not telling you about. So the whole molecular program is not all transcription, but I can't give the introduction unless I focus on a little something. And it ignores all kinds of events and processes. And just for, I'm not sure whom, I'll give you a quick rundown of how we think about this problem. And this also hasn't changed in quite a long time. That clock and cycle, clock and B mal in mammals, drives the transcription of the messenger RNA negative regulators. They are translated in the cytoplasm into proteins. Those proteins associate, and then post-translational regulators, phosphorylation, acetylation, et cetera, come into play that modify those proteins in the cytoplasm. Those proteins go into the nucleus. They directly interact with the transcription factors, suck the transcription factors off of DNA, if you forgive the verb there, and then the negative regulators are subject to a degradation process, which then liberates the transcription factors so that they can rebind to DNA and the process can begin again. And that's probably 85% accurate, and in my world, that's pretty good. So, some of this will turn out to be not completely as I described it, but pretty good. So again, I think Ravi has given you a sketch of this, but I'm really impelled to say something broader about the problem than clocks. Why do circadian rhythms have such a broad reach? Why am I standing here? Of course, I would be standing here giving a seminar if I hadn't won this thing, so maybe I shouldn't put it that way. So circadian rhythms have their fingers in everything. There is nothing in human physiology or pathology, I think, which is left untouched by circadian rhythms. Some of it is profound, and some of it may be a little peripheral, but I think there's, if you look in the literature or you talk to people, there's really nothing which is untouched. I'm always asked, and Fred, you forgive me here, I'm always asked when I show this slide to undergraduates, what about the sex part? And so it's libido, right, which is under strong clock control. And there's probably more, Fred, but right, we're not going to go there. Okay, so that's sort of the processes that are influenced by the clock, and now why, right? So each statement has a subtext, and the first thing to say is that clocks are everywhere. They're in every tissue. This same machine is present all over the body in mice and in flies. And the reason I show this is because I think it is the first demonstration of this general principle, a paper by Jeff Hall and Steve Kay, Plautz et al. In 1997, if I'm not mistaken. And they put a reporter gene that had a regulatory region from a circadian gene and then luciferase. So if that regulatory region is cyclically transcribed, then the luciferase, like the first couple of slides I showed you, would be cyclically produced. And there's the picture of the animal. And then they snapped off body parts and put them in culture, the antenna or the wing or the leg of the fly, and put them in culture. And then the luciferase kept going like this in culture from an antenna or a wing or a piece of fly. So totally separated from the brain or from some central organizing center, and which means, of course, that these clocks must work autonomously in these different tissues to drive the luciferase rhythm and so forth and so on, right? And that kind of experiment has been done in in a mammalian context as well. And this is the mouse equivalent slide, summary slide of the same phenomenon. This is, you guys have seen this before. This is a slide that my pal Uli Schibler gave me several years ago. So the suprachiasmatic nucleus, the SCN in the hypothalamus, was once thought to be the sine qua non of clocks. Without it, you don't have clocks, but now we know this is a much more dispersed system and the clocks are in all tissues. And because of this fact that clocks are everywhere and in a molecular sense have their fingers in all kinds of downstream processes, a large fraction of gene expression in all animals, the mouse experiment from Hognish's lab is illustrated here on a heat map, that a large fraction of gene expression is under circadian control. And this is a picture from the liver. This is 2 days of data. I can't remember if the blue or the yellow is high or low, but it doesn't matter. The point is that for these couple of thousand genes, those transcripts are going up and down every day. And the peak for different genes is at different times of day, which is why, in fact, there's a slope to the curve when you're examining a couple of thousand genes. And if you add up the number of oscillating genes in the liver and the kidney and the brain and the heart and the spleen and skin and muscle, blah, 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 you come to, half or more of the genome. And so this is a rationale, if you will, a molecular rationale for why so many processes are under circadian control. So medical importance of circadian rhythms. This is a list I've been using for a long time that I tweak a little bit when something new appears. And so let me just say a word about two of these things. One is metabolic diseases. To my colleagues here, who are Joe Bassett, et cetera, who are really experts in this. I think it's almost incontrovertible that circadian biology has having an impact on this category of diseases. And there are two papers. I added one last night, you pointed out to me. I added one since our conversation from last night. One paper here from people you all know, Martha, Joe, Fred, about circadian timing of food intake, and then a paper from Sachin Panda and colleagues from 2012 indicating how important food intake is on metabolism, right? And this time-restricted feeding or time-restricted eating has become quite trendy. I do it myself, I must say, not because the evidence is terribly strong, but if you don't eat for a long time every day, it helps you lose weight. You don't have to be a rocket scientist to have understood that connection. So, having mentioned food, I should really say a very important principle here is that food has an incredibly, a remarkable effect on the entrainment of peripheral oscillators. So the peripheral oscillators mean non-brain, right? So only the brain gets light information. We have this unusual black mask which covers our brain, and there's only one way for light to get in, and that's through here. And then you'd imagine that all of this, then the question comes, how then are all the peripheral oscillators connected to this light information which goes here? And the answer is in between, much to everyone's surprise, from experiments about 15 years ago that came from Mike Menaker and Uli Schibler, that food is an intervening entrainment signal. And of course, normally you eat every day at the same time, so you can't tell the difference. But if you feed animals at the wrong time of day, then the entrainment of all this stuff is completely altered, so-called internal desynchrony, and this is a big reason why you feel crummy under jet-lag conditions, because one part of you is ticking at one rate and one part of you is at another phase. So I'll also say a word about sleep disorders and mention the fact that there's this great connection between the Drosophila clock genes or I should say the Drosophila/human mouse clock genes, because we're talking about the same genes, and sleep syndromes. That is, 4 of these genes have been found to be causal for human sleep syndromes, either advanced sleep phase syndrome or delayed sleep phase syndrome. So the field has gone full circle, starting with fruit flies and clock genes, finding the homology with mammalian genes, and then finding that human sleep syndromes, almost diseases, are caused by mutations in these fruit fly clock genes. So, a remarkable turn of events. So, in the last piece of time I have, I want to talk a little bit about a set of experiments that has been ongoing in my lab for a while, and even mention some recent unpublished results that deals with fly sleep. And so, because I knew I would have spoken for 30 minutes on other stuff, I've had to truncate the core of this presentation, but I've listed for you on the left the phenomena which are almost identical between humans, let's say, mammals, humans, and flies. And so I'll simply assert that fly sleep really is very, very similar to mammalian sleep. And the parts of this I want to emphasize is the fact that it's under circadian regulation. So the circadian clock is making you sleepy or alert at different times of day, and therefore reinforcing the drive to sleep, or for that matter, inhibiting the drive to sleep. That principle is true in both organisms. I want to mention the fact that this phenomenon of arousal threshold, the principle is that when you're asleep, the amount of a poke or the amount of noise that it takes to get you to respond is greater than when you're awake. And that's also true for a fruit fly. If the fruit fly is sleeping and you try to poke it with a brush, you have to do a bigger poke to get the fruit fly to respond when they're asleep than when they're awake. Arousal threshold goes up and down with sleep. I'm not going to speak about this, but I am personally very interested in this, I should say, for 2 reasons. One is because I suffer the sleep problems that old people, aging people suffer, namely early morning awakening and multiple awakenings and so forth and so on. Fruit flies do exactly the same thing as they age, identical. I'll simply assert that and leave it there. And lastly, homeostatic regulation. So this is arguably the most interesting part of sleep research, because nobody knows the answer to this question, and it's a big prize if I can use that term from an intellectual standpoint. That is, there's somewhere something that's keeping track of sleep need, how much you've had and how much you need. So when you stay up, when you don't get enough sleep, the next night you sleep longer and deeper, and you're making up for that missing sleep. And fruit flies are the same, except for deep sleep is a phenomenon that's more difficult to assay in fruit flies, but certainly the longer sleep is true. So I'm going to tell you a little bit about some experiments from my lab, and these experiments are due to a fantastic postdoc who just left, Fang Guo, who spearheaded a lot of this work, much published, much unpublished, and he worked with a master's student, now technician, Megan Halla, who's coming here to Northwestern to graduate school in a few months. And so here's the backdrop of this story. So the circadian system in the fly brain is much, much simpler than the comparable system in the mammalian brain. So even if we just restrict our mammalian view to the suprachiasmatic nucleus, the little region of the hypothalamus, which is very important, that has about 10 to 20,000 neurons. They're densely packed. There's a little bit known about, there are a few tools to deconvolute that spaghetti-like sort of mess there. And in flies, there are 75 neurons on each side of the brain, and they're really well separated. There's a half a dozen groups or so of these neurons, and they're very easy to access and to do experiments on to ask the question, what is their function? And in work that we began about 15 years ago, we identified a set of cells here that we called morning oscillator cells, and we identified a set of cells here, for these yellow cells, which we called evening oscillator cells. In fact, for the cognoscenti, that's 4 of these cells and that red one there. And so what is this morning and evening stuff? So it's really quite simple. It is that fruit flies, and for that matter, mosquitoes and other invertebrates, have a very characteristic activity pattern. That is, they show a peak of activity in the morning, coincident more or less with dawn, and they show a peak of activity at nighttime, more or less coincident with dusk. And this is why, if you live in a climate that's, that has a mosquito problem in the middle of the afternoon and the middle of the night, you don't have such a problem. But at 7 p.m., you got a problem. And it's due to this circadian control of their activity. And Seymour, this is a quote from Sidney Brenner, and it's to some extent to frame both our notoriety and also a little bit of what I'm going to tell you. My own opinion is that hypothesis-driven research is in third place. I think you can argue about first and second, but I put first place development of new technology, which really pushes the field forward and allows you to do things. Number 2 is discoveries, which sort of means stumbling on something unexpected and then pursuing that unexpected band on a gel or a little blip on the oscilloscope or something. And last is new ideas. Ideas are cheap, most of them aren't right, and unless they're really linked to experimental results. So what one thing that Fang originated was those tubes I showed you for monitoring locomotor activity. Fang decided that this was just not conducive to doing the kind of modern experiments we wanted, and so he built what we call Flybox, and the essence of this little machine is that our flies are running around in 96-well plates instead of individually in these tubes. And there's food in the 96-well plates, and they actually have much more room to run around than they do in the tubes. And we videotape the flies' activity, and the data match what you get in those tubes, but is actually richer. The data are not constrained by end effects and stuff like that occurs in the tubes. And then as the picture indicates, there are 2 more additional bells and whistles here. One is a red light and green light for illuminating, for activating channel rhodopsins, which are either activating crimson or inhibiting with a chloride channel. So those 2, so we can put optogenetic tools into the fly, and because the animal is, the cuticle is so thin, the light penetrates directly into the brain, and there's no need for all this complicated stuff that you have to do with mice. And lastly, there's a solenoid, and so we can tap the tray and then stimulate the flies so that keep them awake or other kinds of experiments like that. So that's the idea. And this is connected with this is complemented by the incredible collection of driver strains that have produced by Janelia, by Jerry Rubin and colleagues in Janelia, where you can have a piece of DNA which will drive expression even in one single neuron. And we have mined those to get very specific drivers for the circadian neuron subsets that we're most interested in. And so as you'd predict, perhaps, from what I said, that if we optogenetically activate these evening cells that are responsible for evening activity, then in fact, and we turn the light on right here, then instead of having this nice piece of evening activity, here, when there's a little bit of effect you see from the light, even in the control strain, but this is what the pattern looks like in the strain, which is carrying activating channel rhodopsin in those neurons. And if we do the experiment with inhibitory channel rhodopsin, which shuts down neural activity in the same cells, then here's the peak here normally, and now this is what happens in the strain which carries that channel rhodopsin in green light. It wipes out the activity peak because, of course, it's inhibiting the ability of those cells to drive activity. And there's a third set of, let me see if I have it here. Yes. Okay. So there's a third set of cells that I want to talk about, which are in the dorsal region of the brain, and they complement, their function complements the morning and evening activity, and these neurons put the cells to sleep. In other words, when we activate these neurons, and now we're activating them, not inhibiting something, right? That the normal pattern of activity we would see is completely shut down by activation of these DN1s. And so we've come to a model in which there's cells which fire in the morning, there are cells which fire in the evening, and then in between here, especially at siesta time and in the night, these DN1 cells are sleep-promoting cells. They fire and they actually release glutamate, and unlike in the mammalian nervous system or vertebrates, glutamate in the fly, the major excitatory neurotransmitter in the fly is acetylcholine, and glutamate here is working as an inhibitory neurotransmitter with a inhibitory GPCR, and it inhibits the function of those circadian cells. And so this is not too surprising. This is the classic pattern I showed you before, and of course I had only indicated the morning and evening peaks, but now we know that these troughs or valleys are actively promoted by a set of cells which are promoting sleep. And so I want to segue here rather quickly to the connection, to another connection from these sleep-promoting cells, and point out to you, as I said in my introductory remarks, that sleep is under homeostatic regulation, and so far all the loci I've talked about are circadian, and so now I'm going to bridge directly from these circadian neurons to a homeostatic sleep center. And the center that I'm going to describe or try to link to is the ellipsoid body in the central brain. And this is a sleep homeostatic center. Let me see what comes next. No, this is right. So this is this ellipsoid body, which has outputs to these this so-called ring structure, has been proposed. It's been published that this particular structure in the fly brain is very important for driving sleep and is a sleep center. And so the reason I'm telling you this is because it turns out that there's a very interesting and simple connection between our circadian neurons here, these DN1s, and this central complex, or these ellipsoid body neurons, which have been previously shown or proposed / shown, depending on how strongly you believe the experiments, to be a sleep center. And so I'm going to sort of go quickly through experiments, just a couple of experiments that, and sort of how we've gone about it to try to trace the circuit from one place to the other. And I'm going to go quickly because I just want to get to a couple of general principles at the end. And the details here are perhaps of interest to a few neuroscientists, but not to most of you. We use this new tool to try to identify downstream neurons of our DN1s. And it turned out that these DN1s identified the target. Here's the evening cells, which we could identify as a target. And we found sort of a new target that was on the anterior side of the brain, and which is part of the circuit of A to B to C, of DN1s to these TU-BUs, and then to the central complex. And as you would predict, or as the neuroscience people here would predict, if we activate the DN1s, then we should be able to activate these TU-BU neurons if they're bona fide downstream targets and if the connection is excitatory, of course, and we can do that using all of the contemporary tools to try to show that that's really true. And then these neurons are connected to the central complex in exactly the way I described at the beginning. And I'm showing this because this very simple technique is really fantastic. This is expansion microscopy, in which the connection between these TUB neurons and this particular superior projection turns out to go right to the neurons, which are the ring neurons of the central complex. And you can see here that the upper branch, the so-called superior branch, overlaps, in which the purple and green overlap, but the inferior branch is only purple, meaning there's one very specific projection from these TBU neurons, which goes right to the central complex. And then in experiments which, to some extent, recapitulate something that Van Swinderen's lab has published, is when we activate these DN1s, so the circadian neurons, then in the ring neurons, we get calcium oscillations, which actually look a bit like sleep spindles. In other words, the DN1 neurons are supposed to put the animal to sleep. I shouldn't say supposed to. They put the animal to sleep, and then when you activate them, you actually cause oscillations in these ring neurons. And this is the connection from a paper from Van Swinderen's lab, the one I mentioned to Ravi an hour or two ago, that these oscillations, we speculate that these oscillations may serve a similar role has been proposed for sleep spindles, for instance, the blocking of external stimuli. And we can show that this, I'm going rapidly because I said I'm 3 minutes over 45, right? But I'm good, right? Right. When I was an assistant professor, I went to see the dean. It was a long time ago. I went to see the dean, and I said to the dean in the course of a bit of acerbic conversation, I said, listen, you work for me, I don't work for you. So I have a different attitude now that I'm an old guy, right? So this is an experiment. This is an experiment designed to test the relationship between the DN1s on the one hand and these ellipsoid body neurons on the other. And I think I'm not going to show you the results. I'm going to show you the setup, because the setup is sort of more interesting and more accessible for the non-cognoscenti. So here are our flies sitting here, and here's the solenoid, which is tapping the box and stimulating the flies. And so the flies are entrained for 3 days, and then there's a mechanical stimulation, 1 hertz, 10 taps, in other words, 10 seconds of tapping every 30 minutes. So every 30 minutes they're given 10, so it's, you're being woken up every 30 minutes by 10 taps. And this is a record of the taps, because this is the activity of the flies throughout this day when they're being tapped all the time, right? And then both LEDs, the stimulating LEDs and the inhibiting LEDs, are used simultaneously, and the sleep-promoting DN1s have an activating channel rhodopsin in it, and the final sleep structure, the EB neurons, have an inhibiting channel rhodopsin in it. And this is an epistasis experiment to show that if you activate the sleep-promoting neurons, then you change arousal threshold in the way you should, you raise it, but it requires that the EB neurons be accessible to the signal. So if you inhibit the EB neurons, you wipe out that particular response. So that's the result, and I'm going to skip over the actual experiment because I think in a way the concept is sort of more interesting. And so here's the model that we're thinking about, that we have previously shown that these DN1s connect to the evening cells. This inhibits locomotion. And now we have this new connection on the anterior side of the brain here in which we get this structure activated, these neurons activated, increased oscillations, increased firing, and maybe a link here to homeostatic regulation, maybe, and certainly a link to arousal threshold based on the experiments we've done. And I'm going to leave you with one last thought, which I hope you'll find interesting. It's certainly not convincing, but I think it's interesting. And it's the following. This central complex, this region in the center of the brain that I told you is connected with sleep, actually has a richer connection experimentally to vision and to attention. And in fact, it's known to be involved in the processing of visual information from the eye, which in fact is conveyed by these same AOTU neurons, the same ones that we have connected the DN1s. So this pathway and the final landing place, if you like, is used by these two completely different systems and principles, and the question is why. And so here's the speculation. You can read this, right? So perhaps the increase in arousal threshold that we get during sleep shares features, anatomical features, maybe neurobiological features with attention. Because what you want to do, if you want to pay attention, let's say you want to continue tracking, navigating in a particular direction, you would like other stimuli to not interfere, increase your sensitivity to the particular, let's say the particular goal you're tracking. And so the idea is maybe this increase in arousal threshold is being used simultaneously in sleep and in proper attention. And I'm going to end really by just making one political comment in preparation for Congress tomorrow, same one I'll make tomorrow. So this year, including the physics guys, economics, Richard Taylor down south of here, University of Chicago, 8 of the 10 American science winners, 8 of the 10 Nobel Prize winners this year in science were Americans. We're all old guys. A woman in Argentina a couple of weeks ago took me to task for how come there aren't women on this list, and she was right. We're a product of our age. She said the women were home taking care of the babies and cooking, and that's how we could, do great things, and there's some truth to that. But the point of this slide is that we have been the beneficiaries of unbelievable long-term funding from the federal government. And the physics guys are even more remarkable than us physiology and medicine people, because their experiment, their LIGO experiment, seeing the gravitational waves that Einstein predicted in 1916, they started that experiment 50 years ago before having success. And what they published along the way, the things they did, are quite modest. And NSF funded them for 50 years at the cost of $1 billion total cost, $20 million a year over 50 years. And so the point here is that if America wants to have a substantial amount of success in 2037, as it had in 2017, we really have to make sure that our federal government, and to a large extent the public, understands how important this is and how good they have been to us. And with that, I'll close, and thank you very much for your attention. Yeah. So you have some people here on this campus that are so much more expert than I am, but in the interest of time, I'm going to just I'll try and answer that the most important wavelength for daylight wavelength to reinforce your clock in the daytime and to avoid in the nighttime is blue light. And that's why even your iPhone and your iPad has this little app on it to turn the screen orange, so that when you're reading at night, your blue light receptors in your eye are not stimulated. So the idea is blue in the daytime and avoiding blue after sunlight, after sundown.
What about like artificial light?
So again, I'm almost, I'd love to say. So the artificial, we live, this stuff is providing us continuous summertime, but it's not as good, it's not as potent as taking a walk outside, between 10 and 2 in the afternoon, right? So that's the short answer. If we could restrict this to the daytime, it would be better than being in darkness. But having this kind of thing at night, there are circadian biologists who are changing the wavelengths of the lights they use at home on their desk and so forth and so on to avoid, blue light in a desk lamp and so forth and so on. Yeah.
Michael, can you?
Yeah.
Sorry. When cells and tissues have competing cyclical rhythms, and I'm thinking specifically of the conductive system, say from a myocardium, where they have rhythmic depolarization that occurs on the basis of, say, sodium channel dominance. Those cells, of course, still have intrinsic circadian patterns that are influenced by day and night cycles. Is there an understanding about how those two competing processes, say, like calcium, can keep themselves separate rhythmically?
So I think the short answer to that is that under most circumstances, and I'm sure this is true in the heart as well, although I'm sort of guessing based on first principles, that the channels that are involved and the principles are largely separate. So you can have one slow oscillatory phenomena happening in one time zone with one set of molecules, and the rapid stuff that happens 60 times a minute, completely different. There's a few exceptions to that rule. I mean, not in the heart that I know of, but in the cell cycle, where there's some overlap between timing principles, and that overlap is deliberate. Stem cells and skin and so forth. There's a few cases, but by and large, evolution has figured out how these two can live together as bedfellows without competing. Yes?
You mentioned that the eyes are the light receptor that the soul, window to the soul of circadian rhythms here. But it makes me wonder, is there no light perception or contribution of light perception through the skin? And also, what happens with blind people?
Right. So, I think the answer for the skin, and you correct me if I'm wrong here, right? There was a very popular paper that was published about 20 or 25 years ago about the back of the knee, and that was light in the back of the knee, and that was, I think, debunked and perhaps even retracted, if I'm not mistaken. Do I remember? Was it actually never retracted? So anyway, that's not the case, and I think that the light question is still true. Now, the reason I'm smiling is because I have always answered this question about blind people in a certain way, and Phyllis Z, who's a circadian clinician and treats people, blind people, right? That in fact they do get quite reasonably entrained by social cues. That's what she told me this afternoon. News to me, because I have always answered this question in an entirely different way. Namely, if you don't have eyeballs, let's say you lost your eyesight in an industrial accident, then you have no way to get in, for light to get in. I think that's still true, but the social cues perhaps can complement. And there's a dedicated set of, a dedicated photoreceptor and a dedicated set of retinal ganglion cells, which are specific for a specific path to the circadian system. So I had always answered this question with sort of half and half, the ones with IPGRCs and the ones without. And I just learned that I've been giving an incorrect answer. I thought that you couldn't re-entrain with social cues, but live and learn.
In blind flies, that's probably true.
So the fly sight is a little complicated because the clock neurons in the fly, I told you the fly brain is directly light sensitive because of the thin cuticle. And there's a molecule in insects called cryptochrome, and it's a photoreceptor. And it sits within the circadian neurons. And there's also a role for the eyes, but these two pathways complement. And so that's an unusual feature of the fly because of light being able to penetrate directly into the brain without using the eye pathway. Yeah?
When disrupting sleep, it seems like it could be initiating a short-term stress response, especially with tapping on the fly box or shaking someone awake. Is there a way that it's possible to tease out the differences between a short-term stress response and a longer-term circadian rhythm disruption in terms of gene expression?
So it's a very insightful question you ask. Ravi and I have touched on that in the last few hours. What you say is completely true, and it's true for, as far as I know, almost for all sleep restriction paradigms, and it even confounds the classic experiments by Reschhausen here, in Chicago ages ago with the rat, because there is an enormous stress response by sleep restriction, and I'm not sure if you can sleep restrict flies, if you can and avoid that. I think it's an interesting question. How subtle sleep restriction can you do? For all I know, if we the night before coming here, I stayed awake an hour after my bedtime to watch Comey on ABC, and which I regretted because I didn't think it was gripping enough to stay awake. But I was exhausted from just being awake 1 hour too long and then having to get up early to catch a flight. And so it may be that just going to bed 1 hour late with no particular tapping or being bouncing around the walls is enough to produce an endocrine stress response. We just don't know. I think it's those two really are, there is a serious confound there, in my opinion, and I think in yours too. Yep. Yes?
From the description of the signaling circuit, it seems like it's relatively fewer players relative to other regulatory circuits in cells.
You mean, we talking about genes and proteins and things, or are you talking about neural circuits?
Genes and proteins.
Yeah?
Is there a reason why it isn't evolved to be more complicated in certain types of organisms, let's say,
So I got, except for the last sentence when the microphone, for some strange reason, made your voice go softer.
Is there a reason why it seems like the organism has evolved something more complicated to regulate circadian rhythm for some type of evolutionary advantage?
Well, so I don't know of an organism which has something, I don't know of an organism. So plants are more complicated for sure. And their system is more complicated. And it may be because they're sessile and they're extremely dependent on wavelength and time of day and all photosynthesis and all that kind of stuff. They are really more complicated. So I don't have a good evolutionary answer for you. There is no question that it is simple. And I should also add that, this is both a curiosity and was an incredible experimental facilitator for us. The genes, by and large, the genes are inessential. So in fact, you can imagine if you do a mutant screen for fast or slow flies, and your screen is flies that have an altered circadian phenotype, those genes, at least the mutations that you're studying, have to be not lethal to development. And so it's quite remarkable that period and timeless and a whole collection of these genes are really an essential, clock as well in the fly. And so the fly is, and it's a little controversial, there's no longevity phenotype, they mate fine. So to a first approximation, the system is really dedicated to sit on top of everything. And why it doesn't have 75 components. Now it may be that there's a whole bunch of others that we simply haven't mined, but it seems to be the core machinery seems to be quite simple. I mean, maybe it's like Sonic Hedgehog, and that the big players are just a small number of things, and we just haven't teased out some ancillary stuff.
Thank you, Michael.